I want you to try and imagine mapping out every single wire, every microscopic switch and circuit in a massive supercomputer.
Right, which would be hard enough on its own.
Exactly. Just mapping a static system like that would take lifetimes. But now, add an impossible catch to it. I want you to map all those circuits while the computer is, actively building itself from scratch.
Right, soldering its own wires in real time.
Yeah. It's like you aren't just looking at the final product. You're trying to read the blueprint at the exact millisecond the architect is drawing it.
Which sounds like science fiction, honestly.
It really does. But that impossible task is essentially what scientists have just done. So, welcome to today's deep dive. We are exploring a groundbreaking 2026 paper from the journal Cell Genomics.
Yeah, and it's a huge one.
It is. Researchers have created the very first high-resolution, single-cell transcriptomic atlas of the adult central brain of Drosophila melanogaster, which is, the humble fruit fly. But honestly, calling it just an atlas feels like a massive understatement.
Oh, totally. I mean, it's really a meta-atlas. The research team didn't just map a few things. They generated this massive amount of their own single-cell data. And then, and this is key, they integrated it with multiple publicly available data sets.
Right, combining all the knowledge.
Exactly. They essentially took over 1 million cells from the fly head, put them into a solution to isolate the individual cells, and ultimately, they analyzed 329,466 specific neurons from the central brain.
Okay, wait, let's linger on that for a second. How they actually analyzed those cells, because the methodology here is like the foundation of everything we're going to talk about today.
Yeah, we have to define single-cell transcriptomic.
Right, because they didn't just look at them under a microscope, right? To visualize this, I want you to imagine taking a highly complex organ, separating every individual cell into its own tiny microscopic droplet of liquid, bursting it open, and then reading the floating genetic receipts.
The messenger RNA.
Yes, the mRNA. For those who need a quick refresher, mRNA is the transcript. It tells you exactly which genes were actively turned on or off in that specific cell at that exact moment in time.
And the staggering part of this specific paper is the sheer depth they achieved. They got an unprecedented 9.8 times depth of coverage.
Okay, meaning they didn't just read the genetic receipts once.
No, they effectively sequenced the equivalent of every single neuron in that central brain almost 10 times over.
Wow, 10 times.
Yeah, because when you're reading billions of lines of genetic code, you are going to get, statistical noise, errors, flukes in the machine.
Sure.
But by reading the exact same genetic library 10 times, you wash away all that noise. You get this absolute high-fidelity definition of the genetic identity of these cells.
So they can be incredibly confident in what they're seeing.
Exactly. And because of that depth, they managed to confidently identify 4,167 transcriptionally distinct neuronal subtypes.
Over 4,000. That's incredible. Now, I know what you might be thinking. A fruit fly hovering over your kitchen counter might seem, completely disconnected from your life.
Yeah, people always wonder why we study flies.
Right. But understanding how this tiny brain wires itself is like a master key. It gives us the foundational rules for how complex behaviors emerge from raw genetics, how neuroplasticity works, and ultimately how our own human brains develop. Because the fundamental rules of neurobiology, they scale up.
They do. Evolution is conservative. If a biological algorithm works in a fly, nature is going to reuse it in a mouse and eventually in a primate.
So to understand a system with over 4,000 distinct types of cells, I guess we have to start at the actual construction phase, right?
Yeah, exactly.
Yeah.
The central theme of this research is that a neuron's adult identity, what it actively does when the fly is fully grown, is inescapably tied to its developmental history.
Like its childhood defines its adulthood.
Completely. So the brain is built from these neural stem cells called neuroblasts. The entire adult central brain develops from just about 100 pairs of these neuroblasts.
Just 100 pairs?
Yeah. They sit symmetrically on the left and right sides of the developing embryonic brain.
Okay, so I picture each of these neuroblasts as like a distinct genetic zip code. If the entire fly brain is a country, the specific stem cell you descend from is your state.
That's a perfect analogy. And all the neurons that come from one specific neuroblast form a family tree, which biologists call a lineage.
Okay, lineage, got it. But I noticed in the data that these neuroblasts, they don't all operate the exact same way. The paper separates them into type I and type 2. What's the difference there?
So the geometry of how they divide is fundamentally different. A type I neuroblast operates kind of like a solar craftsman.
Okay.
It divides, creates one neuron, again, creates another neuron. It just chugs along and eventually builds a lineage of roughly 200 neurons.
Slow and steady.
Right. But type 2 neuroblasts operate more like a factory manager. There are only 8 of these type 2 stem cells per brain hemisphere.
Just 8.
Just 8. And when they divide, they don't make neurons directly. They create intermediate neural progenitor cells.
Oh, so they hire middle management.
Exactly. They create this amplifying layer. That intermediate cell then goes on to divide multiple times itself.
Ah, I see.
So because of that middleman step, a single type 2 neuroblast can produce a massive lineage averaging over 600 neurons.
Wow, so it's a massive multiplier effect.
Yeah, and what makes this fascinating from an evolutionary standpoint is that this type 2 division pattern, using intermediate progenitors to exponentially multiply the neuron count, is the exact same biological strategy used to build the massive folded cortex of the primate brain.
Wait, really? The fly uses the same scaling trick as a monkey?
Yes. It's nature's ultimate scaling algorithm for computational power.
Yeah.
You just insert a layer of biological amplifiers.
That is wild. Okay, so we have these neuroblasts dividing, creating lineages, but here's a detail I want to unpack. If a parent cell splits into 2, how do the 2 new sibling cells know to be different?
Right. Why aren't they clones?
Yeah. Do they just, flip a coin to decide their fate?
It's not a coin flip at all. It's an unequal inheritance.
Okay, unpack that.
During the birth of these neurons, the cells undergo what's called asymmetric division. Before the parent cell pinches down the middle to become 2 separate cells, it physically sweeps specific destiny-determining proteins to one side of its membrane.
Like shoving all the furniture to one side of the room.
Exactly. So when the split finally happens, one daughter cell inherits all of those specific proteins and the other gets absolutely none.
Oh, wow.
And the key protein in this mechanism drives what is known as notch signaling.
Notch signaling. Okay, so one sibling gets the notch protein and the other is effectively left with a blank slate.
Right, and that creates a permanent fork in the road. That fork splits the family tree into 2 distinct branches called hemilineages.
Hemilineages. Hemi meaning half.
Exactly.
Yeah.
The notch-positive sibling goes down one developmental path, and the notch-negative sibling goes down a totally different one. And the researchers found that these hemilineages end up defining the primary anatomical and functional subunits of the entire adult brain.
Okay, I have to pause here and challenge the implications of this because it sounds crazy.
Go for it.
We are talking about fully mature adult brains here, right? I would assume that an adult neuron is defined by the active job it is doing right now. Like maybe it's processing vision or it's controlling a wing muscle.
That's what most people assume, yeah.
So are you saying that a neuron's identity in the adult brain is still dictated by who its parent was and whether it inherited that notch protein back when it was just an embryo?
Yes. The data proves that developmental history overwhelmingly defines genetic identity, even in the fully mature adult.
That's insane.
It is. When the researchers fed all 329,000 cells into their clustering algorithms, basically asking the computer to group them solely by which genes were currently active, the cells naturally grouped themselves by their hemilineage.
So they didn't even tell the computer about the family tree?
Nope. The computer just looked at what genes were turned on, and it naturally rebuilt the family trees. Because the genetic markers from their birth, specifically these things called hemilineage-restricted transcription factors, they remain active throughout the fly's entire life.
So let's do a quick recap of what we've covered so far, a little bullet summary, because this is dense stuff.
Good idea.
So bullet 1, the brain is built from stem cells called neuroblasts, which act like genetic zip codes or lineages. Bullet 2. These cells divide unevenly using notch signaling to create 2 distinct family branches, the hemilineages. And bullet 3, that childhood zip code permanently defines the adult neuron's identity.
That is a perfect summary.
But if we do the math here, it exposes a huge gap in the logic.
Okay, let's hear it.
We have roughly 100 pairs of neuroblasts, right? They split into 2 hemilineages via that notch unequal inheritance. That gives us about 200, maybe 400 distinct genetic zip codes tops.
Right.
Yet the paper identified over 4,000 neuronal subtypes?
Ah, yeah. You caught the math problem.
So how do we get 4,000 types from only a few hundred zip codes?
The spatial map alone, the zip code, isn't enough to generate that much diversity. We have to introduce the second major axis of neuronal identity.
Which is?
Birth order, time. To generate thousands of unique cell types, the developing brain relies on a highly precise biological clock ticking inside the neuroblast.
A clock.
Yeah, a genetic clock driven by temporal transcription factors, or TTFs.
Okay, transcription factors are proteins that they lock onto DNA and turn other genes on or off, right?
Exactly.
So we are talking about time-based genetic switches, but how does a biological clock actually tick without losing track of time?
It functions through this beautiful biological domino effect. As the stem cell divides, it produces a specific protein, let's say one called MAMO.
MAMO, M-A-M-O.
Right. The concentration of MAMO builds up inside the cell until it hits a critical threshold. And that threshold physically unlocks the DNA for the next protein in the sequence, which is called PDM3.
Okay, so MAMO triggers PDM3.
But here's the clever part. PDM3's very first job, once it's active, is to go back and turn off the MAMO gene.
Oh, it shuts off the previous stage.
Exactly. Then PDM3 builds up, triggers the next factor called BRZ4, and shuts itself off.
Okay, wait, I love visual memory tricks. Let's make a mnemonic for this cascade. MAMO, PDM3, BRZ4. How about many people breathe?
Many people breathe. I love that. MAMO, PDM3, BRZ4. It perfectly describes this self-regulating sequential cascade.
So it creates these distinct, non-overlapping windows of time?
Yes. And the neurons born during the MAMO window get a permanent genetic stamp that is completely different from the neurons born, say, an hour later during the BRZ4 window.
It really reminds me of the dynamics of, like, settling a new frontier.
How so?
Well, the early-born neurons arrive when the brain is essentially just an empty expanse, right?
Right, there's no infrastructure yet.
So they have to act as the pioneers. They have to build the highways, lay the foundational architecture, and stretch their physical connections across these vast, empty spaces. And because they are doing all the heavy lifting of construction, I'd imagine their genetic toolboxes need to be incredibly complex.
That is spot on. The sequencing data maps perfectly onto that pioneer analogy. The researchers looked really closely at a gene called IMP, which acts as a permanent marker for early-born neurons.
IMP for the pioneers.
Right, and they compared that to a gene called DATI, which marks the late-born neurons. In the mature brain, IMP and DATI expression are mutually exclusive.
You can't be both.
Nope. You are either a pioneer or you are a late-arriving follower.
And if those early-born IMP neurons are laying the structural highways, their genetic readout should theoretically look much more complicated than the followers, right?
It absolutely does. Early-born IMP-positive neurons make up a massive 30% of all the neurons in this central brain atlas.
Wow, 30%.
Yeah, and when the team analyzed their total RNA output, these early-born neurons consistently expressed a significantly higher number of unique transcripts and unique genes.
So their toolboxes are physically heavier.
Exactly. They're transcriptionally highly complex because they have to maintain these elaborate physical projection patterns that span all across the brain.
They need a bigger genetic budget just to maintain the infrastructure they built.
Right. But by the time the late-born neurons, the DATI-positive ones, are generated, all that infrastructure is already established.
The roads are paved.
The roads are paved. They don't need a massive toolbox to build highways. They act more like local delivery drivers.
Okay, I like that.
They are structurally more similar to each other. They share overlapping genetic profiles, and they just specialize in filling in very specific local circuits within that established framework.
Let's do another quick recap.
This is good.
So we solved the math problem. If we synthesize this, we end up with an incredibly elegant mechanism for building a brain. You use a grid of space and time. Your hemilineage zip code crosses with your temporal birth minute, the ticking clock, and those two coordinates lock in your exact neuronal identity for the rest of your life.
Space and time dictate everything.
It feels like a foolproof, perfectly deterministic system, but biology hates rigid rules.
It really does.
The most fascinating part of this paper to me is what happened when the researchers found cells that actively broke this spatial temporal grid.
Yeah, the system does break, and it breaks for a very specific functional reason. We call it functional convergence.
Functional convergence. Break that down for us.
So in the clustering algorithms, most cells faithfully grouped with their family tree, just like we talked about. But suddenly, the researchers found neurons from completely different lineages, born in completely different regions at totally different times, clustering together genetically.
Wait, they just abandoned their family signatures?
Yes. They grouped together based entirely on a shared physiological job.
Okay, I think of this like the grueling process of medical school.
Okay, wait on me.
You pull 100 people from 100 different hometowns, totally different backgrounds, different zip codes. But once they spend years becoming highly specialized neurosurgeons, that shared functional identity kind of takes over.
Oh, that makes sense.
They wear the same scrubs, They use the same intense jargon. They hold the same tools. If you are clustering them by their current behavior and uniform, their shared profession completely overshadows where they grew up.
That is a great way to put it. In the fly brain, this functional convergence is overwhelmingly seen in two specific types of cells, neuroendocrine cells and monoaminergic neurons.
Monoaminergic, that's a big word.
Yeah, it basically means neurons that produce specific neurotransmitters. These are the highly specialized cells tasked with producing and releasing complex chemicals like neuropeptides, dopamine, or serotonin.
The heavy hitters.
Right. These chemicals orchestrate global brain states like hunger, aggression, sleep. And manufacturing these molecules requires a massive, dedicated factory of enzymes and transport proteins inside the cell.
So the genetic machinery required to run, say, a dopamine factory is just so heavy that it drowns out the background noise of their developmental zip code.
Exactly. The surgeon uniform, in your analogy, is the genetic sequence for those neurotransmitter pathways.
Give me a concrete example from the paper. How does this translate into an actual brain function we can picture?
Let's look at a group of cells called H-deltour neuron.
Talk, okay.
They are located in the central complex of the brain, which is basically the hub that manages navigation and behavioral arousal.
So this is the navigation control room. What did the single-cell atlas reveal about these specific neurons?
It showed that they heavily express a specific neuropeptide called ASTA. But the single-cell resolution revealed a crucial twist that nobody had seen before.
What's the twist?
The exact same neurons pumping out the ASTA chemical also strongly express the genetic code for the receptor of that chemical, which is known as ASTA-R1.
Wait, so they are releasing a chemical signal, but they also have the biological antenna to detect their own signal.
Yes, it creates a localized, self-regulating feedback loop.
Like a biological thermostat.
Exactly like a thermostat. If the neuron fires too much and floods the local area with ASTA, its own receptors detect the overload, and it can immediately inhibit further release, keeping the behavioral state-like navigation or arousal perfectly balanced.
That is so elegant.
And because this is a brain-wide meta-atlas, they can simultaneously map every other cell type in the entire brain that expresses that ASCII receptor.
Oh, wow. So they instantly revealed an entire hidden network of chemical communication.
Instantly. That level of detail is exactly why this 9.8 times depth of coverage is revolutionary. But the researchers actually pushed the resolution even further than that.
Even further? How?
There's a section of the atlas focusing on a neuropeptide called HUG, short for Hugin. H-U-G-I-N.
HUG? I love that. What does HUG do?
HUG is vital for integrating a fly's internal state like, say, severe hunger with external sensory cues, and it modulates feeding and sleep cycles.
Okay, so it tells the fly when to eat and when to sleep.
Right. And when the researchers isolated the neurons expressing this HUG peptide, the deep coverage allowed them to disentangle distinct functional subtypes that would normally just blur together into one blob of data.
Okay.
They identified one specific subtype called HUG-RG. Based purely on the genetic transcripts, they mapped its specific co-transmitters and its precise receptor profile.
Okay, so they found a highly specific subtype of HUG neurons. How many of these HUG-RG cells are there?
The HUG-RG subtype consists of exactly 4 cells in the entire adult fly brain.
Wait.
Exactly 4.
Just let that sink in for a second to you listening right now. Out of 329,000 neurons packed into an organ the size of a poppy seed, they didn't just find these 4 cells. They read their individual genetic receipts well enough to map their distinct functional capabilities.
Yeah, it demonstrates that we finally have the analytical resolution to identify the rarest, most hyper-specialized neurons in biological systems. We aren't just grouping things by general brain regions anymore.
No, we are defining the individual cellular components of behavior.
Exactly.
Okay, so let's pull all these threads together for our final recap. We started with the concept of a self-wiring supercomputer. What these scientists have done is basically translate the original source code.
They have built a definitive bridge between development and adult function. They've proven that the developmental programs, the asymmetric divisions, the ticking temporal clocks, they aren't erased once a brain reaches maturity.
They stay there forever.
Yes. They serve as the permanent foundation of adult capabilities. And if we can understand the universal rules of how the fly brain diversifies its circuits in space and time,
then we can apply those exact same algorithms to vertebrate brains.
Absolutely.
And this is why this matters to you. The exact same mechanisms that build those 329,000 neurons in the fly are driving the development of your own nervous system.
Yeah, humans use these same principles.
Every time you recall a memory or feel a wave of exhaustion or even just navigate a complex intersection on your drive home, it is governed by an architecture built on these exact principles.
And this Fly Atlas, by the way, is fully public. Anyone can explore it at flycns.com. It's a foundational step toward understanding human neural plasticity and mapping the genetic roots of neurological diseases.
We have moved past just mapping anatomy. We are now reading the developmental history and the functional destiny of every cell simultaneously.
It's a whole new era of neuroscience.
It really is, which leaves us with a pretty profound concept to mull over as we wrap up this deep dive.
Wait on us.
We've spent this time exploring how a fruit fly requires this unfathomably complex cascade of stem cell divisions, unequal protein inheritances, and temporal domino effects, just to allow it to eat, sleep, and fly.
Right.
So if that level of developmental choreography is rigidly required for a tiny insect, what does the hidden developmental map of your 86 billion human neurons look like?
That's a staggering thought.
And even deeper than that, how much of exactly who you are today, how you process the world and how you feel, was already pre-programmed by the exact millisecond your very first brain cells were born?
Wow. It really forces you to rethink the boundary between raw biology and your actual identity.
It does. Something to ponder the next time a fruit fly lands on your kitchen counter. Thanks for joining us, and until next time, keep diving deep.